Okay, well, my name's Scott D. I'm with the Pipestone Group. I was a professor at the University of Minnesota for 12 years at the vet school. I was a practitioner for 12 years prior to that. I'm the research director at the Pipestone Holdings Company. So Pipestone is made up of several different divisions. There's a veterinary services group. We've got 6 clinics, 40 vets across the Midwest. That's where my research division, Pipestone Applied Research, is located. We take care of what's called the Pipestone system, which is now about 75 managed sow farms for family farmers. And it's about 250,000 sows across those 75 farms, and we'll be at 300,000 by the end of the year. So I think we're number 3 on the list. So that's management from breed to wean. We've also got management from wean to finish through our Grow Finish, Nutrition and Records division. We've got a marketing division, we've got a processing plant, and we have a China and Mexican division. So that's what I was talking about over there, was the Chinese experience with African swine fever that we've actually have felt ourselves. It's not a good feeling. But today we're talking about something different, and this is a study we published this year. We did it a couple years ago, so I actually had to go back and read it because I was a little bit foggy in some of the details, but I went back and looked at it. And you might wonder why you would do this. And you think back a few years ago, there was a lot of talk about ABF production, but there really weren't any data. There wasn't anything published that you could say, well, if you have ABF production and you get a disease challenge, what happens? And so we partnered with Zoetis to do this study. And so there's a citation listed there in +1. Let's see. It's on the side. Oh, the side. Got it. All right. So there's a quick overview of our group. I already talked about that, so we won't do that again. Our research division, you can see our objective. We're an applied research division that I oversee. We've also got a contract research group. We've got 5 wean to finish research facilities. These are 2,400 head wean to finish facilities that producers use. I mean, basically representative of what's in the field. However, we've modified them for research. We've put in pen scales, we've put in feed logic system, etc. I'll show you a picture. The study that I'm going to talk about was conducted in one of those. We've got a new BSL-2 facility we just built, so we can do some really nice side-by-side treatment and control work. And then we get funded primarily, similar to Dr. Plummer, I heard him talking about industry support. We've got a group of like-minded systems across Asia, Canada, and the United States that pay us to have our research information. So they pay us a monthly fee that goes into the budget. We partner with companies like Boehringer, Merck, Zoetis, PIC, Topics Norsvin DNA. We do trials with them, competitive trials, basically looking at different vaccines side by side with control groups, an experimental challenge, or sire line trials looking at offspring performance from different sires through the harvest facility. So it's a lot of fun working with those companies, and they pay us to get into our facilities. And so even though we lose money in almost every one of those trials, it's a way to bring some funds into the company. We like controlled research in applied conditions. So we like challenge models. You can see the challenges we've conducted in our facilities there. And we kind of look at our work in 3 big buckets. One is more competitive testing. I've described that. Vaccine A versus B, genetic line XYZ. We also do a lot of validation of processes. So when PED hit and we wondered how do we sanitize a contaminated trailer, we developed a model to understand that question. And then a lot of the work I do is proof of concept. If you're familiar with some of the transboundary work we've done, looking at how viruses can move around in feed around the world, or how PED can be transmitted through feed, some of that initial work that was done at our group. So that's kind of where I spend my time. Okay, so here's research barn 1 through 5. They all look the same. They're basically got 2 rooms, split down the middle. The attic and the pit and the room are all divided. 2,400 head, about 84 test pens and some off-test pens. Again, feed logic system to measure feed use, and floor scales for taking, and as well as individual scales. So they're just like barns that are used out in the field. And that was one of our goals, is let's recreate the model that our customers and shareholders use, so the data are applicable under large numbers of pigs. So the study you'll see here is 700 pigs in a group. So you start to get some nice power when you can put that together. Here's our BSL-2. We just put it up, as I mentioned, this fall. We're running some feed mitigation trials right now, looking at viruses in feed. Can we mitigate that risk? So we've got different rooms. You see kind of how they're set up. We can put in sows, weaners, or growers, so we try to make it as flexible as we can. So just a little background on the company. Any questions? Pretty fast, but gives you a little understanding of our scope. Okay, into the trial. So we heard the title. There's our hypothesis. We're really looking at a couple different things, looking at production performance, looking at welfare aspects and personnel responses in clinically affected pigs raised under ABF conditions versus those under responsible use conditions in the face of a PRRS virus challenge. Our hypothesis was quite simple, that clinically ill animals will require antibiotics. The methods, we source, all these projects start at one of our sow farms. And so our management team, our research team goes into the sow farm, randomly select the animals across the treatment groups, get them tagged and organized. If they need to be any processes performed at the sow farm, like you'll see here, that happens there. Then the pigs are weaned into our research facility. In this case, it was number 3. So we started this from one of our sow farms that was negative to PRRS virus, so no vaccine and no wild-type virus, a truly naive population. This was a Zoetis project, so they wanted to use the Fostera PRRS vaccine given at weaning per label. The challenge, we've developed, in this case, a 10% cedar pig challenge model. And so we'll infect approximately 10% of the pigs in the wean to finish barn and let the virus roll through the population just like it would under natural conditions. And I've grown up a really nice 174 PRRS virus challenge at South Dakota State in Eric Nelson's lab, where we can actually have a very representative strain, the most representative strain today of PRRS virus in the nation, and use that as our challenge. You can see our TCID 50. And then the route of administration. We spent a lot of time on this talking to experts in welfare, talking to producers and veterinarians who have raised ABF pigs. We have a welfare division, but we wanted input from scientists. And so we talked to a lot of really smart people that have worked with ABF, several, Anna Johnson, for example, at Iowa State, who's got doing some welfare research. We wanted to get some information because we knew this could really go south really, really fast, okay? And we ran it through our IACUC. We ran it through the Zoetis IACUC. So we had the animal care oversight. We had our welfare director involved, Dr. Carissa Odland, who heads up our welfare team. We set a guideline collectively that if in the ABF group, should we attain a total of 20% based on either removals, i.e. treatments with an antibiotic, the pigs were removed from the study because they were no longer ABF, or death. If we got to 20% collectively, we were going to terminate the ABF component. We were going to continue the trial, but we were then going to be able to medicate with antibiotics that ABF group. So that was our line in the sand, so to speak. And never having done this before, maybe we would do that differently, but this is how we did it. Here's the design. It's pretty straightforward. 3 groups, roughly 700 pigs in a group. 3 different antibiotic approaches. Treatment number 1, you'll see throughout the slides, we call that full program. This was the Zoetis designed program. So they said, well, we want piglets to receive exceed at processing and weaning. So 100% of the pigs get processing and weaning exceed. We could use antibiotics wean to finish in the water or the feed. Then they designed their feed plan. Our suggestion for group 2 was a bit of a modification. We said, well, we'll use exceeded processing, but we don't want to use it at weaning. We'll use water medication, but we don't want to use feed medication. So we changed it a little bit. And then there's the ABF group. So the ABF group was like a never ever. So nothing in lactation, nothing in post-weaning. So group 1, group 2, group 3. Now we'd probably do it a little differently. If we did this again, we designed this several years ago, we probably wouldn't use Exceed. We'd probably use something like penicillin, not a critically important antibiotic. So going forward, we'd probably change this, but just for the sake of discussion, this is how we did it, because at the time we designed it, this was fairly status quo. So let's just get right into the results. So here's the Farrowing house data. There's group 1, so that's the full program, treatment 1. Treatment 2, again, is a modified, and treatment 3 is ABF. So the number of litters, you see the enroll weight with the superscripts, the wean weight, so we start to pick up some differences in the ABF group already, in the lactation, and the average daily gain, and the percent removals and mortalities. There was a diarrhea issue in the Farrowing house. There was E. coli involved. We could medicate the first 2 groups, but we could not medicate other than anti-inflammatories. We could use anti-inflammatories, but we could not use antibiotics in the treatment 3. So already in the Farrowing house, in the first 3 weeks of the study, we're starting to pick up some signals of how this might turn out. So this is kind of interesting lactation data. So the pigs go to the wean to finish barn. We inoculate the cedar pigs with PRRS. We start letting her roll. For clinical assessment, we use the Zoetis IPC scoring system that you're probably familiar with. There's A pigs that are early infection, early clinical response. B pigs were kind of in the middle. And then C were more chronic. And so every day, or 3 times a week, we had trained members of the research team scoring the facility, scoring all the pens, and looking for counting up the A pigs, the B pigs, and the C pigs by group. So that's numbers of animals on the y-axis. The x-axis is by time, obviously, and there's group 1, 2, 3 in each of the months, and these are the bars are the cumulative across that month. The total number of A pigs, the total number of, that's in green, the total number of B pigs in yellow, and the total number of C pigs in red. So you can already see in the October, which is prior to challenge, differences across those groups clinically. And then you can see when we began our challenge, which I think was November 3rd, you can see things really took off. And you can see very quickly, we got to a point where we had already exceeded our threshold, or met our threshold of 20% removals, i.e. antibiotic therapy of ABF group, or mortality. So we terminated at that point, as we planned, and mass medicated the entire facility. So now we could use antibiotics beyond that in all 3 groups. So we technically didn't have an ABF group anymore. But it was going to be a welfare issue. We had to. That was our decision we made collectively, and our welfare team was there guiding us. You know, once we kind of treat them, things settle down pretty good. You can see the clinical scores over time. Okay? We did a lot of diagnostics just to see what's involved besides the PRRS virus. You know, what else was in the complex, right? And so there you got pre-challenge and post-challenge. You got flu, mycoplasma, PCV, PRRS, of course, and then anything else we'd find on routine diagnostic submissions. And so you can see there were some other things brewing and cooking along the way. Some mycoplasmas, a lot of rotavirus, and of course, even some flu. Circo. So it was kind of like what you'd see in the real world. You've got a complex here of infectious agents working together. We submitted a lot of tissues, and this is just a collective list of bacteria that were recovered from those submissions. It's kind of like diseases of swine. We basically found everything in there that you'd think you'd find. So we even found Salmonella cholera suis. I couldn't believe that. But you can see many, many bacteria. The primary cause of mortality was PRRS virus and Haemophilus parasuis. That was the most common diagnostic observation. But we did find quite a plethora of bacteria. Now, we measured performance because, remember, the original goal was to try to get some actual data on this ABF process in the face of a disease challenge. And would the use of responsible antibiotic use, would that make a difference when those pigs could be treated with antibiotics? And so you'll see several graphs that look just like this. So we've got the metric on the y-axis, in case here it's average daily gain. We've got time periods of weights that we're taking. We basically take a weight whenever we change feed. And so we broke it up, day 0 to 28, that was pre-challenge, 28 to 49 is post-challenge, et cetera, et cetera. You've got group 1, ABF in blue, no, sorry, not ABF, full program in blue, group 1. Modified program is in red, and then the ABF is the yellow one. So it's not too hard to pick that one out, and you see the differences statistically between the groups. And so not seeing a lot of difference between the first two groups, but obviously a significant difference in gain in the second group, third group. Here's feed conversion, kind of the same picture. Big differences. You probably noticed the typo. That should be an A. Really nothing statistically between treatment 1 and 2, but a clear difference between treatment 3 throughout the rest of the project. So that's feed conversion. This one is a pretty significant graph. This is final market results. And so looking at full value pigs by group, looking at defects, light pigs per group, and then mortalities and removals combined. Okay, so we combine those two together. And you can basically see a big difference in full value, and you see a big difference in mortality and removal. We almost got to 60% mortality and removal in that ABF group. I'd never seen so many dead pigs. We did some other things too. We tried to measure some welfare aspects. We'll kind of highlight a few of them here. We looked at huddling, or pigs piling basically, during November there, during the period of challenge basically. You see all the groups were affected. They all were piling, very feverish, very uncomfortable, as expected. Some numerical differences, and they all got a little bit better after we medicated, but they were definitely affected. We looked at a common industry audit as well to try to see if there were differences in some of the parameters in that metric. If you look at the bottom line, we didn't really pick out a lot of numerical differences. I think that's because we were removing so many pigs from the ABF group because of welfare issues. We pulled them off the study because we had to treat them or they died. So we ended up with really nothing different numerically on the bottom line, but if we had included those other pigs, I think we would have had a big difference in that treatment group 3, but for welfare reasons we couldn't do it. It was interesting, if you look at lameness, how the lameness spiked, which is right here, lameness in each of the columns. You see this lameness spike right after the PRRS challenge. A lot of Mycoplasma rhinus and Hyoscinoviae recovered from Iowa State during that time. So the PRRS virus really flared up, as we know, the lameness issues in these pigs. So that was an interesting observation. We talked to the workers and we asked them several questions to kind of get their perspective on this thing. And let's show you a couple of slides. The question was, are you satisfied with the care provided? So for the workers who are actually able to treat or not treat pigs, what was their level of comfort or discomfort? And you can see this is obviously the ABF group, and high distress is like up here, low stress is here. And so this worker was, the workers we surveyed were very distressed during the ABF period of time because they could not treat their animals in contrast to the other groups where they could. And so they clearly had a problem with the inability to appropriately medicate like they know how. So that was quite telling. And then at the other question, one of the questions, did this study meet the goals? Did we answer the questions? They agreed there. 1 was yes, strongly. 4 would be disagree. Was treatment effective? Antibiotics effective? Strong yes there. And was the time productive? Did we learn what we needed to know? And again, they were very on board. They were uncomfortable though early on. We did a little bit of economics. This is just a simple net revenue calculation. You see the math that we selected for the analysis. You could put any numbers you want in there, but you kind of see the net revenue estimate from treatment 1 to 2. Not a lot of difference there, but a big difference. So if you're going to do this ABF, obviously you better get a lot of money when you sell these pigs if you do have a challenge such as this, because you could see the net revenue is about $70 less for group 3. So to discuss the strengths and limitations and then open it up for discussion amongst yourselves if you like. I like the design. I thought the design was good. We might do it differently, as I mentioned now, but at the time I thought it was representative. Population size was good. The isolate was relevant. We tried to represent the field and the facility and the environment. We had great statisticians. The late Bob Morrison did the stats for us, and then Deba Modi did the stats for Zoetis. And they basically sit down ahead of the study and decide what analyses they're going to do. They take the data independently and analyze it, and they come back together and see what they saw. And so this, as you can imagine, was pretty straightforward. That was good. Multiple metrics from animals to humans. And then it was published. We actually achieved our goal to get it into the peer-reviewed literature with some data that actually says this can happen should you have this situation. Limitations is only one strain of PRRS. That was a very virulent virus. It may be different with the less virulent virus. One replicate, really with just one 6-month turn. It's hard to replicate that. And we don't really have a true negative control here with a non-challenged ABF. So the conclusion was pretty straightforward. As we wrote in the paper, the results of the study clearly indicate that the responsible use of antibiotics is essential to maintain proper swine health, performance, and welfare in the face of a disease challenge, and kind of bring it down, and boots on the ground approach is when pigs are sick, they need to be treated with antibiotics. That's the project, very briefly, to be respectful of your time. As I mentioned, it's published, it's on open access, and so I didn't know if you had any specific questions about it, or if I got through it okay where it made sense.
So, Scott, what do you think if you wouldn't have a challenge there with PRRS? Do you think the ABF would have been much different?
Yeah, good, Lucas. Good question. We've done several others without a challenge, and there are some differences, and not nearly as drastic as this. But there is significantly higher mortality, slightly slower gain. The thing that really spikes the ABF pigs is the modified live PRRS vaccine. That fires up lots of bacteria. So even with that preventative process in the ABF case, if you can't treat those pigs, they get buzzed just simply from the modified live PRRS vaccine. So yeah, we have done this again, just we haven't done it to this degree. But yeah, we do see differences across. And that was in the face of no challenge. That was a high health situation. Rick?
So, Scott, help us understand, in the production environment, are there systems that are trying to go ABF for marketing reasons?
Yes.
And are they successful?
Hard to say.
Yeah, because...
Hard to say.
Yeah, because the economics don't work.
Right. I haven't seen any real hard data. Okay. They talk about these situations, so they are out there. They try to build in isolated areas, but they run it. I think this happens more often than we hear about.
Right. Okay.
Any other questions for Scott?
We just thought this was important to get into published literature, so questions like this can be asked and discussed.
Well, we get two different types of questions. Questions from within our industry on the financial viability of the programs like that, and then questions from outside our industry is like, why do we need antibiotics anyway? And then, so it's two different perspectives.
Yeah.
It's because it's a good data point to handle both of them.
And the welfare kind of ties them both together, and so that's why we wanted to have some, they're very rudimentary levels, but there's some level of welfare assessment, because this was a welfare situation, big time.
Yeah.
Yeah.
For the guys that try to tackle no medication, what do you guys figure is the dollars that the packer needs to pay in addition to make it?
I don't know. To be honest with you, I don't know. You know, this was we saw a $70 difference here. This is extreme.
Well, yeah, I'm just talking about healthy pigs.
Yeah. I don't know. My guess is it has to be quite significant.
That's a percentage.
Scott, would you be brave enough to show these to a bunch of regular consumers?
Sure. That's why we,
Science out of that and show them
Sure.
the effect, you know,
Yeah.
their effect you would have.
yeah, we took videos of this, but we haven't shown them for obvious reasons.
A little too graphic.
A little too graphic, yep. I mean, there'd be 50 dead pigs a day. That's where we were in this deal before we jumped in and mass medicated. It was something.